Hey guys, how are we doing? I am not using a video today, so your computer's working fine. I'm just not using one right now. We are going to go ahead and go over the muscular system, we'll go over the first part of it, muscle system 1, right now. So without wasting any time, let's go ahead and get started. A couple of small corrections here. Sorry, let me get my pen going. This should say three types of muscle cells. Okay, so yes, you've got skeletal muscle, smooth muscle, and cardiac muscle. Cardiac muscle, as you probably infer, is only found in the heart, and it is unique because cardiac muscle has kind of a combination of traits from skeletal and smooth muscles. Okay, so speaking of those characteristics, let's go over this. Skeletal muscle has a tubular shape. Doesn't even look like tubular, does it? Let's try that again. Okay. Are they striated in appearance? Yes. Skeletal muscle is distinct because of its striations or stripe. Skeletal muscle also has multiple nuclei along the cell. Okay. This muscle is voluntary, meaning we control that movement. And its attachment is found along the skeleton. So where you find it in the body, muscles attached to the skeleton. Okay. Then we have smooth muscle. Smooth muscle cells are made out of, they're kind of spindle shaped. So like it says, it has a narrow cylinder and the ends are kind of pointed. Are they striated? No, they are not. It is smooth muscle, and it looks smooth compared to the skeletal and the cardiac muscle because both of those have striations, whereas smooth muscle does not. Smooth muscle has only one nucleus per cell, and it is involuntary, meaning we don't consciously have to tell our smooth muscles to do anything. So things like your blood vessels, your internal organs, your lungs, the muscles that control your breathing, all of that's an involuntary process, right? You don't have to sit there and think, digest my food, digest my food. It's all the smooth muscle in the organs that do that. So blood vessels and organs. Okay, again, our cardiac is a mix of both of these characteristics. It has a tubular shape like skeletal muscle. It is also striated, though the striations look somewhat different. But if you still can't tell if you're looking under a microscope, if you can't tell if you're looking at skeletal muscle or cardiac muscle, you should also look at the number of nuclei per cell because cardiac muscle only has a single nucleus. Okay. Obviously, this is involuntary. You're not consciously making your heart beat. And you find it in the heart wall. Okay? All right, so speaking of skeletal muscles, there's lots of functions. Some of those include, first off, support. Without our skeletons, we would not be standing upright. We'd be a pile of mush. Okay? Also, movement of bones and body structures. Okay. So the skeletal muscles assist in smooth muscle processes like breathing, and then obviously moving at all is done by skeletal muscle because we have to choose to move. Movement of fluids in the cardiovascular and lymphatic system. Shorten that. So like that. Movement of blood and lymph fluids. So we'll get into that, especially with blood, a little bit more as we get into the circulatory system. Okay, protection. Oh, you know what, guys? Did I miss this? Oh, I totally did. I wrote it in the wrong spot. Ugh, fine. I'll just rewrite it. Movement of blood, lymph fluids. Let's see if I can get rid of just. There we go. All right, this one, if I paid more attention, is about releasing heat. So this has to do with maintaining a constant body temperature. So that heat comes from the fact that our muscles are constantly converting potential energy to kinetic energy, so stored energy to energy of motion. And when that happens, whenever energy changes form, it releases heat. It's part of the, warm up your muscles before you exercise type thing. Release heat, kind of loosen them up a little bit, make them a little more willing to stretch without injury. Okay, then finally we have the protection of the internal organs and also joint stabilization. So while it's true that most of our intestines and kidneys, things like that, most of them tend to be beneath the rib cage and the abdomen, a huge number of our organs are held within the rib cage. Not only have the heart, we have the lungs, the liver's tucked up under their stomach. There's a variety of things that are kind of within that protective cage of the ribs. And frankly, even lower down the abdomen with those floating ribs, there's still some of that protection there. All right, let us continue. Okay, so if we're going to look at kind of the structure of muscles, we have this kind of tiny to big type arrangement. One of these little circles in here, okay, so there's tons of these. So if we look at just one of these little tiny circles that are represented are called myofibrils. Myofibrils are the part of the muscle that's going to contract. How about I try to write that so that we can read it? Sometimes this little board is so hard to write on. You should learn that myo, when you see it, refers to muscle. So a lot of the terms that we'll be going over have this as a prefix, or it's part of the word itself. There's another one I'll show you as well as we come up to it. Okay, so we have a bunch of myofibrils that make up the larger muscle fiber, okay? So group of myofibrils equals a muscle fiber. When you have a bunch of muscle fibers, all of these, you end up with something called a fascicle, which is this next biggest part. And then when you have a group of these fascicles, then you end up with something called the muscle belly, which is this big part they don't really have labeled. Okay. Then we have tendon. Tendon is actually a connective tissue. And its job is, as you can see here, to connect the muscle to the bone. Okay. Okay. Let's go ahead and move to the next. Okay, I know there's a bunch of terms here, but hang in. And again, here's that other kind of root, sarco, that I told you to watch for. When you see sarco, you also know you're looking at something that has to do with muscle. Okay, let's do some definitions. So the sarcolemma, which is this peeled back part right here, okay? The sarcolemma is the muscle fiber outer membrane. Muscle fiber and muscle cell can kind of be used interchangeably. Okay, so it's the membrane that's around this whole muscle cell. Then we have our myofibrils. And these are bundles of something called actin and myosin. We'll learn more about those soon. Or actin and myosin, however you want to say that. Because you can say it both ways. The myofibrils contain something called sarcomeres. Okay, so this right here is one myofibril. All right, so a sarcomere is where contraction happens, where muscle contraction. Remember we talked about myofibrils on the last page, and that myofibrils are where muscle contraction happens. Well, it's because they contain these sarcomeres, which are the actual site. Okay. The sarcomeres, as far as muscle contraction, they're going to shorten. Okay. All right, let's keep going. Now let's look at the sarcoplasmic reticulum. I know it sounds like the endoplasmic reticulum, which you see in animal or other types of cells in the body. Here we're looking at the sarcoplasmic reticulum, and this is something that's going to store and secrete calcium, which is going to be needed to make the muscle actually move, as we'll see in a minute. The sarcoplasmic reticulum is this kind of blue web, veiny looking thing. It is wound throughout the muscle cell itself. Then we have our T tubules, which are these green guys right here. Okay, the T tubules transmit electrical signals into the cell. Signals that are going to be sent from the nervous system. What they actually are is little invaginations in the plasma membrane. This term, invagination, you can see in a variety of different places in biology, usually referring to a cell or a cell membrane. As here, the plasma membrane, it kind of just means that if you're looking at a cell from a different angle, this is like an invagination. Okay, and it's a little pouch, pocket, or indentation in the membrane. Okay, so let's talk about our actin and myosin. Myosin is thick. These are thick filaments. Whereas our actin is thin. Okay. Myosin is stationary. It does not move. Okay. So when a muscle contracts, it's not the myosin that's going to move. It's actually going to be the actin, and it's going to slide inward. So this is like a random example, but if you think about yourself walking along flat ground, but you have like a railing next to you, and you reach out and you grab the railing, and you use the railing in your arm to pull yourself forward. Okay. In that example, the railing is the myosin. It's stationary. It's not moving. Your arm is the actin, where it's shortening to pull you forward. Okay. All right. Again, a lot of vocabulary. Hang in. You do need to know it, but hang in anyway. It'll be okay. All right, this is one of the first big processes that we're really going to go over in a little bit more detail in this class. Cellular respiration was a big process, but for this class, it gets very, very abbreviated. So when it comes to the physiology part, this is kind of one of our major process lectures. Before I go on and explain the steps of muscle movement, I would suggest that you become familiar and comfortable with the terms that I've given you in the past pages, okay? Especially the page that we just did. So you can pause this video for a minute, go back, read through it, identify those components on the image that's on that page. Just give yourself a minute to familiarize with the different parts. Once you feel like you have the vocabulary down and can kind of picture that image in your mind of where everything's placed, then you can move forward and start learning these steps and start memorizing the steps. If you try to memorize the steps without having any comfort with your vocabulary, you're going to struggle. That's going to be hard. Okay? All right. Not struggle because this is the hardest thing in the world. Struggle because it's hard to know what you're talking about if the vocabulary is so confusing for you. All right, having said that, I actually very much enjoy the process and the efficiency of what happens in muscle movement, just like I enjoyed it with the sodium potassium pump and the nervous system. I like all that it goes into this as well. All right, let's review action potential. That's from our nervous system. Remember that the action potential is a signal or a stimulus coming through the nervous system to relay that something needs to be done. So the action potential in a neuron is sent to the effector, which in this case is going to be a skeletal muscle cell. Okay, so back to the put your hand on a hot stove. The brain is going to send a signal back to the nervous system saying move your hand. That action potential gets sent to the effector, the skeletal muscle cell, and the cells and the arm will contract to pull the arm back. So action potential then is conducted from the nervous system to specifically the motor neuron. The motor neuron is the one that's going to send that last action potential to the effector. It does this by secreting something called acetylcholine. Okay? When the motor neuron releases the acetylcholine binds to receptors that are on the sarcolemma. Remember that the sarcolemma is the membrane around the muscle cell. Okay? Now we've got to get that signal inside the cell. Okay. So the action potential travels along the sarcolemma, the muscle membrane, and then travels down into the membrane through our T-tubules. Remember that these are the little invaginations or the little indentations in the membrane. So once the action potential gets into the cell via the T-tubules, it's going to release calcium. Which part of our muscle cell had the calcium stored? Let's see, sarcoplasmic reticulum. Okay, going back up to the top. Do it again. Action potential is conducted from the nervous system to the motor neuron. The motor neuron is going to release acetylcholine, and at the end of the axon terminal, that will stimulate the skeletal muscle by binding to the skeletal muscle membrane called the sarcolemma. Once it binds to that part of the sarcolemma, the action potential then is taken into the cell via the T-tubules. The presence of that action potential in the cell triggers the release of calcium. That calcium has been stored by the sarcoplasmic reticulum, and the sarcoplasmic reticulum is the one that will release the calcium. Calcium is a positive ion, and when it's released, it floods the interior of the cells to find our sarcomeres. Okay? All right. On the actin filaments, remember this is the part that moves during muscle contraction. On the actin filaments, there's a protein called tropomyosin. Tropomyosin covers the binding site on myosin. Okay? The protein that's bound to the tropomyosin is something called troponin. Calcium is going to bind to the troponin, which will then pull away the tropomyosin. And it's going to expose the myosin binding site. At that point, I'm going to finish this part off and review it. the myosin heads, the binding site, will bind to the actin filaments to form something called a cross bridge. Okay? When this happens, the myosin, I'm going to put this in quotes because it doesn't really pull it, but the myosin pulls inwards in something called a power stroke. Okay, so pulls our actin in. All right, how fun was that, you guys? Look at all those terms. All right, don't freak out. So I want to go back here to the other half of step 2, where we talk about the tropomyosin and the troponin. Basically what you have is, you have your actin, right? This is the short fiber or the short filament that's going to move in our muscle contraction. And on that, there's this gate, if you will. And the gate needs to be open for the myosin to attach to the actin. Well, the gate is covered, and it's not only covered, it has a guard that go with it, okay? So we have tropomyosin, which is like the gate itself, and then we have troponin, which is like the guard. When calcium binds to the troponin pulls the tropomyosin away from the myosin binding site. So I want you to picture a guard standing at a gate, okay? And then the guard is going to get a message, which is going to be calcium. When the guard gets the message, he opens the gate, okay? If we put it like that, it's kind of simple. Guard at the gate, guard gets a message, guard opens the gate, okay? Again, our guard is the troponin, the gate is the tropomyosin, and the message is the calcium, okay? All right, so once the gate is open, now we can have the myosin bind to the actin in something that's just called a cross bridge, which you can see in the image right down there at the bottom. And yes, ignore those green triangles. But you can see the cross bridge between the actin and the myosin. And then the myosin, quote unquote, pulls the actin inwards as the actin shortens in what's called a power stroke. And there you have your contraction. Okay, as always, I can't see your faces to know if you're partly in tears, bored stiff, a really good mix of both, or if you're fine with all of this. Once again, look over the vocabulary here, try to get comfortable with it, and then you can start breaking down the steps. I found in teaching this that a really, really effective way to help you remember this for your exams, and frankly, once you get into phys classes, is if you draw this out yourself. So if you sit down to study and get yourself a blank piece of paper or two and draw this from action potential all the way through the power stroke and label it, it will really help you remember what's happening and why. Okay? All right, let's move to this next page. Oops. Which I believe is the last page. All right, so here's the thing. We've had all of these steps happen for our muscle contraction. We've made it through the power stroke, but now we need that muscle to quit contracting. We need it to let go. All right, let's talk about what we do. First off, we need ATP. Remember ATP from cellular respiration, the energy source? Well, ATP is required to make this contraction process stop. So ATP binds to the myosin head and detaches it from the actin filament, right? So we had our cross bridge thing happening where we had our myosin head, we had our actin attached, but now we're going to have ATP come in. ATP. And it's going to bind to the myosin instead and kick out the actin. Okay, so it's kind of like ATP is the new girlfriend and she kicks you out, or the new boyfriend, however you want to look at it. Okay, ATP binds to the myosin head, detaching it from the actin, making the actin let go. Okay, so ATP becomes ADP because it's part of it's a phosphate that's going to do this. So ATP becomes our ADP, and that gives it the energy to cock the myosin head back into this power stroke position. Okay, so basically, like I said, we had the myosin head attached to our actin filament. ATP breaks up this happy relationship. When that happens, we've got the actin head the myosin head and the actin filament back in this position where it's ready to form another cross bridge and ready for another power stroke. So it resets it so the muscle can contract so that the head can reattach to actin again to start another cycle or another contraction process. Okay, so the sarcomere will continue to shorten. Remember that the sarcomere is inside the myofibrils and contains the actin and myosin. It will continue to shorten as long as the motor neuron is sending action potential. Okay? When the signal stops, when the action potential doesn't get sent anymore, calcium ions are going to get actively transported back into the sarcoplasmic reticulum. I'm not going to be able to fit it. Reticulum. And at this point, our gate is going to be slid back into place. So our tropomyosin. And it's going to cover up those myosin binding sites again, which will allow the sarcomere to completely relax. Okay, let's go over this part again. We've just finished a power stroke. In order to get the myosin and the actin to let go of each other, ATP comes in and disrupts that by binding to the myosin itself. The ATP is converted to ADP because the energy is used to make the myosin and the actin let go of each other. Once the actin and the myosin have let go of each other, they're in a position to once again form a cross bridge for another power stroke cycle. Okay? This process will continue to repeat as long as the motor neuron keeps sending the signal to do it. Once the signal is finally stopped, calcium gets actively transported back into the sarcoplasmic reticulum. Actively meaning this requires energy because just like we saw with the gated channels in the nervous system, calcium does not want to go. It's crowded in the sarcoplasmic reticulum, crowded with other calcium ions, so it has to be forced back. When it's forced back, remember that it was the messenger or the guard. So when that message is taken away, our gate closes, and tropomyosin closes and covers up the myosin binding site again, and the sarcomere stops contracting. Okay, I know this is crazy redundant, but I'm going to do this. I'm going to talk it through one more time because I'm not going to rewrite all of it. But follow along with me. And frankly, if you are good at this point and don't need to hear me walk through this one more time, feel free to turn this off. For those that need to Get it solidified one more time. Here we go. All right, so we have our action potential, our signal that says that our brain is telling us that we need to do something. That action potential, sorry, scratchy voice. That action potential is conducted, again, from the nervous system to a motor neuron. The motor neuron secretes something called acetylcholine. The acetylcholine binds to receptors on the sarcolemma, which is the muscle membrane, muscle cell membrane, okay? Once the acetylcholine binds there, it allows the action potential to travel along the sarcolemma and into the membrane, through the membrane, into the cell itself, through a T-tubule. When the action potential reaches the inside of the cell through the T-tubule, it triggers the release of calcium from the sarcoplasmic reticulum. The calcium ions flood the interior of the cell to find all the sarcomeres. Okay, here we get to our messenger and our gate. On the actin filaments, we have this protein, our gate, called tropomyosin. Okay, and it's covering the binding site where actin wants to hook up. So then we have our guard, troponin, and it's bound to the tropomyosin. It's holding on to the gate. Well, when the troponin gets the message, which is calcium, the calcium is going to bind to the troponin, to our guard, and the guard is going to pull open the gate, the tropomyosin. Now those myosin binding sites are accessible. And so then the myosin heads at the binding site will attach to the actin filaments to form something called a cross bridge. Then the myosin, quote unquote, pulls the actin inward for a power stroke, just like our contraction. When that needs to end, ATP binds to the myosin head to break up the relationship between the actin and the myosin, okay? The ATP is converted to ADP in breaking up this relationship, which puts the actin and the myosin back into a position to start another power stroke. The sarcomere will continue to shorten as long as the motor neuron continues to send the potential, the action potential, or the signal to do that. When those signals finally stop, the calcium is actively transported back into the sarcoplasmic reticulum, and the tropomyosin, or the gate, once again covers up the myosin binding site, and the sarcomere can relax. Okay, now, how come stiffs are stiff? If you've never heard that slang before, a stiff is a dead body, okay? And the other stiff, how come stiffs are stiff, is referring to the process of rigor mortis, which is the body essentially freezing into place, locking into place after death. And the time of rigor mortis setting in depends on all kinds of factors, from temperature to humidity to cause of death, all kinds of things. But it's usually within the first few hours up to the first 12 hours, and then it can last for another 12 to 24 hours. All right, so how come dead bodies go through rigor mortis? Well, I want you to think for a minute about ATP, because that is the answer to this question. I'll give you the answer in a second, but I want you to pause this if you need to, because I want you to think through it. Let's think about ATP. What process creates it in our bodies? So cellular respiration creates ATP, right? All right. What do we have to have for ATP to happen? Oxygen and glucose, okay? Well, if a dead body is dead, it's not breathing, which means there's no oxygen. If there's no oxygen, we don't have any ATP being produced. All right. Now, what does ATP have to do with our muscles? Well, ATP has to break up the relationship between myosin and actin. When it does that, the contraction can release and reset. Okay. If there's no ATP, we can't break up that relationship, which means the contraction continues until the fibers eventually wear out. That is why stiffs are stiff. There's no ATP to make the myosin head and the actin filament detach from each other to stop the muscle contraction. So it simply stays like that in the body until eventually the calcium and such starts to break down on its own. Okay? Alrighty. If you have questions on this, please message me. Otherwise, I'll be back shortly with the other half of this.